Good afternoon, everyone. So yeah, my name is Jason King. I work for Qian, and Qian is a WuXi advanced therapies company. WuXi Advanced Therapies is based in Philadelphia, US, of course. So starting with a little bit of good news, of course, it's really good to see how many developers there are in the cell and gene therapy space. It's even better to see how many new therapies are making it all the way through to market approval. The slight issue we have, of course, is that the pricing on these therapies is extremely high. So at Qian, we're looking at what we can, which is the viral vector production platforms that we've developed in-house to see whether we can generate stable and scalable forms which have the potential to reduce the cost of goods, at least for the viral vector manufacturing. And we would then hope that that would filter through to the end prices that are being paid. So if you look on the left-hand side, let's see if I can get this to work. Okay, so here, these are the standard systems that we use, which are transient systems for producing AAV and lentiviral vectors. So they use plasmids, which means that they're pretty flexible, but they're not very scalable above 200-liter bioreactors. So what we did was, for lentivirus vector production, we created packaging and producer cell lines for lenti. These, I'm glad to say, are as productive as the transient systems. However, because they use either less DNA or no DNA, the cost of goods associated with the input costs is far lower. On the AAV side, we created TESA technology. And TESA technology doesn't aim to reduce the input costs, but has aimed to increase the productivity of the system. So I'm going to talk about these two systems very quickly today. The first one is the lenti system. And of course, for lenti, we take a traditional approach, which is stable cell lines. And you can see on the left-hand side that if you want to make lentiviruses quite quickly, you simply take hex cells and transfect in 4 different plasmids. The packaging cells are containing 3 of those 4 components, which means that you only have to transfect in a single plasmid, the gene of interest or the therapeutic transgene. You can take this one step further to the producer cell line by incorporating everything into the cell line, which means you don't have to transfect that to produce lentiviral vectors, you just need to induce with DOCs. So you can see top left, what we actually did was to take hex cells, introduce the GAG-POL and VSV-G components so that their expression is inducible. The REV component, however, we included in 2 different ways. So one constitutive and one inducible. And you can see from the graph below that the recovery of pools was very efficient. The gray line at the bottom is a negative control. And this is just looking at the productivity of the pools initially. So that's between 2 and 3 times 10 to the 7 transducing units per mil for the constitutive and inducible REV cell lines. So we took the best performing pools and we cloned them out. So don't need to look at the details here. The point is we analyzed over 460 clones for each cell line. We analyzed them on the basis of their transfection efficiency, but also the infectivity of the lentiviral vectors that are produced from each clone. So overall, it was a massive improvement in these clones compared to the pools, and some of these clones were more efficient at producing than the full transient system. So we selected the top 20 clones for each cell line, and we expanded them further for testing. So top left, you can see the first 5 bars are from the constitutive clones, then the next 5 large bars are from the inducible clones, and you can see above that dotted line, we've got a couple of clones from each, which are producing at between 2 and 2.5 times 10 to the 8 transducing units per mil, which is really high. On the right-hand side, you can see that we've been looking at the stability of these packaging cell lines. It's an ongoing process, and these error bars on passage 11 are overlapping, so it looks pretty good. And of course, we're testing the cell lines in the presence and the absence of antibiotics. Ideally, we want to have cell lines that don't need any antibiotics at all in the process. So the data I've shown you top left is introducing a GFP expression plasmid into these packaging cell lines. The data at the bottom is asking the question, well, what about other therapeutic transgenes? So here, the blue bars are the packaging cell line being transfected with a single plasmid. The red bars are the 4 plasmid transfections using the same GOI plasmid. So you can see, in all cases bar 1, the packaging cell line is superior to the transient system. So that was packaging cell lines. Now we move on to producer cell lines. So the producers are created using the transposon system. We do this in order to increase the copy number of the gene of interest. And we initially did this with a HEC293 cell line that WuXi has a lot of experience with. And you can see down in the bottom left, we were able to come up with clones which were producing at 2 × 10⁸ transducing units per mil upstream. We were acquired by WuXi Advanced Therapies, and that gave us access to another HEC293 cell line, so we decided to apply the same process to that. So we're currently at the pool stage. We haven't cloned them out yet, but you can see from these titers that even the pools are producing at about the same level, or one of them's actually twice the level of the clones from the earlier cell line. So now, using these stable pools, we generated producers, again using this array of different potentially therapeutic transgenes. And you can see that the titers now are sort of mid-E7. So 5, 6, 7, 8 times 10 to the 7. One of them's just over 10 to the 8. So this is looking very good. And of course, these are pools still, so it will be possible to subclone and do process development to raise these titers higher. This is a summary slide for the lenties. So you can see us comparing the packaging and producer cell lines to the 4 plasmid transfection on the left-hand side. The benefit of the producer cell lines, of course, is that they don't need any plasmid at all, so the cost of goods is far lower. Variability in production is lower, but the productivity is in the same order as the transient system, which is really good to see. And this is also the case for the packaging cell lines, so the productivity is the same for the packaging cell lines. The cost of goods is slightly higher than the producer lines, of course, but the packaging cell lines are useful because you have flexibility. You can transfect in any gene of interest you wish to package. I should say that there's a master cell bank in process. We expect that to be ready in Q1. That's for the packaging cell line. For the producer cell line production service, the version 1 cell line is available now, and the version 2 one, which you see looks pretty promising, that's going to be Q1 next year as well. So that was Lenti. Changing gear, this is Tessa, onto AAV. So for Tessa, we didn't make cell lines, we looked at nature and we learned from nature. So in nature, AAV is produced at high levels, high quality, through the addition of an infectious adenohelpervirus. The problem with adenohelperviruses is that, as well as supporting AAV production, they also produce a heck of a lot of their own capsids. So you see in this electron micrograph, the nucleus is completely stuffed full of adenovirus fiber protein. So what we wanted to try and do was engineer the adenovirus to prevent it making all of these adenocapsids when we really want to make AAV. And we did this, it's shown on the right. So you see this is the adenogenome. So we've got one promoter expressing all of the late genes, which is really convenient. So we just had to engineer one promoter. So we inserted into it a tetrapressor binding site, and downstream of it, a tetrapressor expression cassette. This means that whenever there's any expression from this promoter, the tetrapressor is expressed, the tetrapressor binds to the tetrapressor binding site and prevents any further expression from that promoter. However, we need to be able to propagate these adhelperviruses, and we do that using DOX. So when you add DOX to the system, DOX binds to the tetrapressor, prevents it from inhibiting the capsid expression. But of course, when we want to use these as helperviruses, we simply don't add DOX. Of course, the system self-silences. We get early gene expression, but no late gene expression and no contamination. And this is how we use the system. So we use a pair of TESA advectors. One of them has the AAV rep and cap constituents. The other one has the ITR flank gene of interest. We infect normal HEC293 cells with an MOI of 3 for each of the input viruses, and we produce just AAV. And the amount of AAV we're producing per cell is up to, and sometimes beyond, 1 million. So that's 6 particles of adeno in to get 10 to the 6 AAV out. It's a good amplification. And it works for all of the serotypes we've tested so far. So you see the blue bars are TESA, the gray bars are the transient system, and you can see there's about a 10-fold improvement across the board. This is in adherent cells, but it also works in large scale in suspension culture. So you see in the top left, this is actually from a 50-liter bioreactor, and it's AAV6, and we see a 10-fold improvement in upstream productivity. Down at the bottom, this is from a 200-liter bioreactor, AAV2, and here you see actually it's a 20-fold improvement going from the plasmid system to TESA. Of course, you can do the downstream processing as well, purify the particles, and have a look at their quality. And it's interesting to note that for AAV6, 1 in 40 particles was actually infectious. For AAV2, it was even better, 1 in 17 particles infectious. So very high quality particles. So in this summary slide, we're looking at the whole process in blue from left to right, and we're comparing the TESA process to plasmid-based production for AAV6 and AAV2. Interestingly, the TESA platform allows us to generate preps which have 95% full capsids. For AAV6, for example, upstream, 66% of the particles have full capsids, right? This compares to about 10 to 15% for the transient system. So it's much easier to enrich when you have higher quality particles to start with. The take-home message really is that from a 200-liter bioreactor you see on the right, we can produce much larger yields with TESA than with the transient system. And I'm often asked about the input material because the input material here for TESA is more complex than plasmids. But if we look at the top process, this is producing using plasmids. If you were to use a 200-liter bioreactor to produce plasmids, this would be enough plasmid to run 15 to 25 200-liter bioreactors of AAV production. If instead you use a 200-liter bioreactor to produce TESA vectors, these vectors would be enough to run 50 to 100 of these 200-liter AAV bioreactors. So we're able to run more bioreactors, and as you've seen before, each bioreactor is producing 10 to 20 times more material. So with TESA technology, we're not trying to aim to reduce manufacturing costs per batch. However, we're producing a lot more material, so the cost per patient dose should be significantly reduced. So what we're doing now for customers is, at Oxgene in Oxford, we are creating bespoke TESA vectors carrying customers' genes of interest, therapeutic transgenes. And for more and more customers these days, the interest is in synthetic capsids. And so we're producing TESA rep caps for their specific capsids. Larger scale process development can be done in Philadelphia or in China. And finally, we make the GMP grade TESA vectors and subsequently the clinical grade AAV either in Philadelphia or in China as well. The final thank you slide. There's a lot of people involved in this. We're not going to go into each person's name, but we've got 7 teams in Oxford, shown on the left, who are involved in this work, and 4 teams in Philadelphia. So, thank you.